Hello and welcome. This is Columbia Basin Research's second video in a multi-part series on using TagPro. TagPro is a program created by Columbia Basin Research at the University of Washington School of Aquatic and Fisheries Sciences. TagPro is an acoustic tag data translation utility for your desktop that takes valid acoustic tag events produced by fast or equivalent software and creates an output file of capture detection histories used for survival analysis by Atlas or other third-party software. Today in this second video, I'm going to show you how to define your release groups, sites, and runs, and also how to execute your runs. Alright, so first, fire up the program. After loading your data, you can select the Releases tab to define the releases. The top section, labeled Available Release Groups, shows all releases from the tags input file as defined by release location, species, and tag year. The bottom section shows the releases defined by the user. You can define releases in one of two ways. Number one, select one or more available releases and press the bulk add button to add them to the releases window in the bottom section. Number two, select one or more of the available releases to combine into one release. In this case, the user can accept the default name, release names are hyphenated, or edit the release names in the release name box and press create. Selecting bulk add allows you to create a large number of release groups where each line represents an individual release. Selected groups are highlighted in blue. Added releases appear in the lower releases window and are available for further processing in TagPro. Alternatively, you can combine releases into larger groups. Here, rather than treating the wild and hatchery tag groups as individual releases, two releases can be pooled into one by selecting them both in the Available Release Groups section. The default group name can be simplified. Below, we change the combined group to RI tail race all species, and then press the Create button with the result here. I can select the Sites tab to define detection sites. The top section, Available Arrays, shows the locations available as defined in the node's input file. I then define sites in the same way that releases are defined as described previously, in that each line can be created as an individual site using bulk add or pooled by highlighting multiple sites and using create. Here, I've selected the first three arrays, RIBZ, RIFB, and RIDF. Pressed bulk add and then selected the final two arrays, CBAR and SLND. Renamed the site final and pressed create. Once I've defined the releases and sites, I can define the output runs of detection histories. Runs are defined by, number one, the desired output format, Atlas or Standard, number two, the selected release or releases, and number three, the selected sites. To define a run, you must go back to the Data tab and press the Add Run button, bringing up the Add Run Definition dialog. You can give the run definition a name, TagPro example in this case, and select an output format, Atlas or Standard. In this current case, I've selected Atlas. You can then press Next. The Next button brings up the Release Group Selection dialog. Here you can select one or more release groups. In this example, I've selected RI tail race all species. I can then press Next for the Sites definition dialog. Here I've selected all sites. After pressing the Next button, the run definition summary appears. At this point, you can either press back to modify the run definition or finish to complete the run definition. The run is then displayed as shown here. Once runs have been defined, the final step is to select the desired runs, individually or multiple. Each run will be saved individually. Here I've defined two runs and selected both. I can then press the gear icon to process the selected runs. Alternatively, select Run under the File menu. When the runs are processed successfully, the number of records written to the output files is recorded in the status area and a run complete message appears. The resulting output files are placed in the specific output folder. The output file names are defined by the name of the runs followed by the format of the run. The following shows the names of the required fields in each input file. The input files are comma-separated value files, CSV. The following required file names must be in the header of each file, but they can be in any order. Other fields are allowed to be present, but they will be ignored by TagPro. For the tag CSV file, the italicized field names are output as part of the standard format, but not the Atlas format. They are optional, and if present, empty values are allowed. The field names are followed by their data type. TagPro can produce two types of output files. The Atlas format produces the required input format for program Atlas. It contains a fixed number of columns. The standard format has one row for each tag, and unlike the Atlas format, has a variable number of columns depending on the number of detection sites. The columns for tagger, name, bucket, etc. can be used to easily create optional attribute files for program Atlas. Now I will talk about how to save a CSV file in Excel and preserve the seconds date time field. Here you can see an Excel worksheet with three date time fields selected: tag_date, activation_date, and release_date. I can right click on the selected fields and select Format Cells from the context menu. Here we can see the resulting Format Cells dialog. Select Custom under category and type or enter mmddyyh:mm:ss and then press OK. Once this is done, when you save the CSV file, the seconds will be saved with the selected date time fields. This has been lesson 2 on using TagPro, in which we covered defining your release groups, sites and runs, and also how to execute your runs. Please note that if for any reason you're having trouble with TagPro and these videos are not sufficient to answering your questions, you can find the user manual at cbr.washington.edu/analysis/apps/tagproapp. The PDF manual is there, and you can either load that into a web browser as I'm doing right now, or you can download it to view in Adobe. If you're using TagPro and you have any questions or comments on that same page, you may also give us feedback via a user satisfaction survey. We would greatly appreciate any feedback you have in helping make future versions of this program to be more useful to our users. We hope that this video helps you get the most of TagPro. Again, if you have any questions or comments in regard to TagPro, please fill out our survey as previously detailed. You can also contact Columbia Basin Research at web@cbr.washington.edu. Best of luck on your analyses, and thank you.